it's here.
Started. So good morning all of you. Again I welcome you all on this third day of our GIN program and hope you are enjoying the sessions. So we have kept the lecture in that consequence that, so yesterday we had the discussions on the natural products, how we can isolate the natural products, how you can how you can know about their structures, their different kind of functionalities, most importantly characterizations. So we talked about it. Now I'll little bit of the synthesis part I'll be talking, just to give you some idea that how the scientists were involved in developing new natural products and all, followed by the process research. Like you cannot design, designing will not be helping unless you synthesize them. But you will not be talking in detailed synthesis, but the techniques, okay? So to start with some natural product synthesis, it is called target oriented synthesis. As I said, once you know the structures, you need to achieve that particular goal. So accordingly, you can have your substrate, your different kind of reagents what are available commercially, to do some process to apply the different kind of synthetic techniques to reach up to that product. That's what it's called TOS, that is a target oriented synthesis. Though that time in 1890, these sort of things were not there. They are just going for the molecules and is synthesized. And you can see the first synthesis from that Emil Fischer, it's done on 1890 for the synthesis of glucose. That's a very beginning, early beginning of it. But it is not the isolations. Earlier we were talking about the last day about the isolation of it, but it is the actual synthesis of that particular compounds. Followed by, you can see in the Robinsons, Robert Robinsons, they all awarded the Nobel Prize for that. There are many facilities, but still they could get the corresponding products. After that, the R.B. Woodwards, like he's a stalwart on that particular part for the total synthesis of a wide variety of the natural products. Now here you can see how many different natural products they are targeted, isolated from the persons. Maybe the same person is also isolating them from the nature and targeting them and to get the synthesis done of that. Sometimes maybe the synthesis might not be up to the proper stereochemistry. There are many important factors also were associated with those. But you can see the amount of work and how vast and how wide it is. So Sir Robert R.B. Woodwards, like he was awarded Nobel Prize in '65 for this kind of like total synthesis. These are some representative examples, but apart from that, there is huge. Followed by Professor E.J. Corey in Harvard University. They have also done they're actually like totally their work on the synthesis of the natural products. Retrosynthetic analysis, they have devised the synthesis in different modes. So here from another era started where the natural products were targeted, then they had done all sort of disconnection approach. How you can cut the bond, which kind of bonds will be just breaking and forming, this sort of like process was done on it, and they can come up with again a huge amount of natural products, huge variety of natural products. So in every cases they have developed new methods, they have achieved those things, all cases the retrosynthetic analysis things was working, okay. So that's their immense contribution in the field of natural products, synthesis, retrosynthetic analysis. They are awarded the Nobel Prize on that, but it is Nobel Prize is also not enough for them that how much contribution they have done on that particular time. That time like people has to think like that, not to have a designing, but how to target, how to go to like achieve those goals. That was the main objective for them. Now coming to the target oriented synthesis, we have to know the structure of the products, then you can do all sort of disconnection approach, that which particular carbon hydrogen bond, like carbon carbon bond or carbon heteroatom bond should be synthesized from a particular substrate. So that was the approach. Accordingly, you can go back and find out what would be your starting material to achieve that product. So that was the retrospective analysis. You have to go back and find out where from you will be actually starting. So that was the beginning, because your target is fixed. You need to achieve the goal. This is the natural process you have to synthesize. Then you have to go back to that and get the choose the actual substrate in order to get that particular natural process. So these are some sort of, like you can see the synthesized compounds, how that target oriented things are coming in a very simple way. This compound is synthesized, that is the target, and you go back, this sort of arrow is the retrosynthetic, like for the arrow which represents the retrosynthetic steps. So go back and find out the very simple substrate will be going in a forward directions to reach the target. That was the idea. It's not signaling. Now you can see the retrosynthetic, just a representative examples of that apothiones, how you can devise your tools to reach the target. Now you can see how complicated the molecule is. So this in between, that is the natural products. Now you are devising your synthetic tools in a sequential ways, which step will be going, and all four, there are I guess 5 different kind of retrosynthetic things you can do in order to choose the proper substrate where from you can start your synthesis. So this way people have devised their synthetic, the synthetic tools or the approach in order to target that thing. So there might be multiple options, but the ultimate goal is same. You can take different road to reach over there. You need to choose which way the reagents will be more convenient. It might not be giving some side products. Yields will be more. Maybe it will be taking less time of it, not a multiple steps. Like sometimes the total synthesis is about 100 steps, maybe 50, 60 steps are also there. So you have to concise your method in such a way that in a very appropriate way, choosing the reagents, maintaining their different properties, stereochemistry, you can reach up to the goal with all the properties or parameters should be in a convenient situation. Okay, so that's way the synthesis. I'm giving only one example for it, and I can understand that might be very complicated to understand for you if you like this. But still, I'm not going to talking about any synthetic techniques, but it is just a understanding that how you can have the disconnection approach and to reach from a different substrates where from you can start your synthesis. Is it some problem happening? Okay. So now why it is important? Why the target oriented synthesis is important? Of course, you can have a, you know the compound and you know their sort of biological activities which you discussed before. Now you can target it and you can achieve it. That's a really one-to-one approach. Like you start from there, you can have the proper like choice of the substrates. Therefore, you can achieve your goal. Efficient lead optimizations, which was the beginning of these discussions, that you have to optimize the lead, which will be actually going to work in the biological systems. That's the medicinal chemistry part. Accelerated the drug developments. You can accelerate it. Like if you have a synthetic tool in a very smart way, you can have lot many molecules you can synthesize based on the structures whatever you are having. Natural products, small molecule synthesis expands their drug possibilities. You can explore different kind of biological studies with those drugs. Also, like personalized medicines you can get, like you can just modify that whatever the things you are synthesizing, you can modify from the substrates. Sustainable manufacturing, definitely it will be important. But you need to understand one thing, it's a linear synthesis. You are going A to B to C, something like that. And you are for fixing your target at the beginning. Then you are going back and just making it in that way to achieve that goal. This way you are making your synthetic steps in such a way that you will be getting the ultimate, like whatever your main product, in a very convenient substrate and start, like the reagents and those methods. Okay, so these are basically the importance of it, but again it's a single like linear steps, or maybe sometimes it is a divergent way you can get the corresponding products. Now I'll be focusing something which will give our libraries of compound. Target oriented synthesis, you fix the target, you design the substrate, you figure out the synthetic routes, what are the reagents you will be using, and you can have the target achieved. But if you want to make a library of synthesis, things are different. You need to have a huge number of molecules at the end of the synthesis. So that's a synthetic tool you have to devise. That's what it is called combinatorial library synthesis. So as you can see here, it's a linear steps it is going. You apply your disconnection approach, you apply your retrosynthetic tools on it to go back for the substrate, and then you can start your synthesis. But if you want to achieve, so we'll wait. Okay. They said the streaming is stopped in between. See, this presentation I don't need, you can do it from here. I'm talking about the next one, this is difficult. That is actually the problem. These are the problem actually. Wait. So it's done. Yeah, so there are some technical issues. Okay. So as I was talking about, once you are targeting the total synthesis, that is a target oriented synthesis. So you are going through a linear steps, you are doing, but before that you have to get a proper retrosynthetic analysis in order to know the substrates, reagents, their particular type of like route, synthetic route to reach up to that way. But again, that particular synthesis is a linear and will be reaching to that particular target. On the other hand, if you want to go for a combinatorial library synthesis, like library means it's a huge number of molecules will be synthesized. Accordingly, you have to devise your synthetic tools to reach to that particular diversity. Okay, so you have to create in such a way, you have to start in such a way that it is not only a single compound, you can reach to a multiple number of compounds by using that particular type of synthetic routes. So into that particular type, diversity oriented synthesis is coming into the picture. Now, what is the need? Because as I said last session also, your natural product may not be serving all the biological requirement or the whatever the medicinal requirement up to that level. You need to modify it, and if that total synthesis is extremely important that ways, but if in order to go for better biological activities, you need to have a multiple molecules, a library synthesis will be required to reach to cater different kind of biological activities. That's a bridging between a synthetic chemist to a medicinal chemist. So that medicinal chemist will be proposing different molecules, but you should have a synthetic tool in order to achieve those molecules. It should not be that theoretically you can have this many molecules will be working against a particular protein, but if you don't have the molecule or those compounds, small molecules in your hand, that will not be actually serving the purpose. Okay? So this way the technique should be developed, the process should be developed in order to have a wide variety of compounds in a very concise ways. So this is the way you can have the diversity oriented synthesis. So you can see the difference. If you start from here, a complex molecules, the corresponding substrate will be very simple. You are going back to it and start from the substrate. But here, it's not retrosynthetic, it's a forward synthesis rather. So you can select that one with this substrate, how many different diversity you can create. So you should have the idea in mind that you want to create the diversity, what will be your substrate. And those permutation combinations things will be important in order to get a few million of compounds using your synthetic tools. It is not about a single this one, but you have to get a huge number of compounds utilizing the diversity oriented synthesis. So coming to it again, you can change the different reagents afterwards to get a similar kind of reactions, but at the end, a wide variety of products will be forming. Here also the same thing, you can have the combinatorial library synthesis and the diversity oriented synthesis. Now diversity in terms of what? Diversity in terms of the reagents, different kind of reagent will give the different products on that, or it might be a common reagent, but you change the functional group or you change the some part of the substrate, then also you can create the diversity. You can create the diversity in terms of putting them in the functional group diversity, different kind of appendage diversity, building blocks diversity you can create over there. Along with that, you can create a stereochemical diversity. As you can already talked about that, the stereochemistry is a very important parameter in order to cater a particular biological like activities and these things. So you should have the diversity in the functional group, you can have the diversity on different kind of appendages, building block diversity, stereochemical diversity, and most importantly skeletal diversity, scaffold diversity. So your synthetic tool should be up to that level in order to do all sort of diversity in the final products. Okay, so with that we need to talk about the stereochemical aspects, because stereochemistry is very important parameter to change different kind of biological property of the final product. So to go for it, you can see a very simple example of the limonene, S-limonene, you only the changes in the stereochemistry. You look at it, this is the S stereochemistry and that is the R1. So these sort of stereochemical changes are actually changing the property of that compound that is existing, that is there found in the lemons, and the other one is the oranges. But only the difference is the changing in that particular chiral center. The orientation of the groups are actually changing. Now there are lot many drugs, but the drug molecules may not have a stereo center. That is also possible. So all the cases, the stereochemistry is not that much, means it is not a primary factor, because your molecule may not have a stereochemistry. That is also possible. But if it is there, you have to have proper understanding, and you should be able to correlate the structure activity relationships of the stereochemistry with their final biological target. So this way you can see these are the very common drugs where the stereochemical factor is hugely important in order to cater their biological activities. So now start with what is actually the problem. Now you see that if it is there, you can have a shake hand with the catalyst or the biological target with your small molecules. You have to put your hand in a particular orientation. Otherwise, you cannot shake the hands with them. Okay, it's just a hand shaking. You can have the molecules which is actually going to interact with the target. So, but if you don't put the hand in proper orientations, that hand shaking will not be possible. So that's the main things. Now you can see that thalidomide, I will come in more details of these structures. It's cure the morning sickness. On the other hand, if you vary the stereochemistry, it is causes the birth defects. So only to change the stereochemical output of it, there are huge impact on their corresponding biological activities. So these sort of case studies are done where that naproxen treat the arthritis pain, whereas the R1 cause liver poisoning. So these sort of diversities are there if you vary the stereochemistry of the final product. So that means your stereochemical outcome will be very, very important. Here also you can see the propranolol and the ACE of this one less potent, 40 to 100 times more potent than the R1. So these kind of stereochemical aspects are hugely important in that. Sometimes the drugs are synthesized and administered as a form of a racemic compounds, but maybe the other enantiomer may cause some difficulties or maybe not at all biologically active. So why to administer more drug, more number of molecules in that particular receptor? There is no use. So it should be very much pure in terms of their stereochemical aspects. You cannot make or you cannot have the molecules in a mixture of isomers, mixture of basically the enantiomers. You cannot administer a racemic compound on that. So that's what the efficacy. One enantiomer may be more active than others. Then the other enantiomer should be separated either by some doing, that resolutions of the enantiomeric mixture, or you need to get rid of the other enantiomer, or you have to make your synthesis in such a way that only one enantiomer will be your products. So these aspects are very important and should be keeping your mind at the beginning once you are targeting drug molecules or a library of molecules towards a particular receptor. So this way it has been seduced, and that is a very tragedy what happens to that. This thalidomide was initially prescribed by doctors and given by the medicinal chemist for the nausea effect, like sometimes the morning sickness and nausea. These particular things have been prescribed, but at the same time there was death of the child when they are about to born, like the newborn baby were about to were dying because of the other enantiomer of the same drug. So that was a huge tragedy in all the words, and that you can see the timeline of it, and it was discovered, it was pharmaceutical company was actually synthesizing. But if that was prescribed or that was administered of both the enantiomer, there is a massive destruction or massive effect was there. So that's a real tragedy of it in terms of one stereo center can cause many death of the small childs. Okay, so here from you can get the knowledge and you can have the proper understanding the importance of the stereochemistry. Now there are some other example. You can see this is a marketed drug, omeprazole. There's a marketed drug which is having the ACE enantiomer. Now there is a sulfur which is not actually the chiral, but you can keep it. That particular stereochemistry you can induce on it. That is the omage that is commercially available, that you can get in any kind of market, the very easy cheap drug. But if you can have in an enantiopure form, their efficacy is increasing, right? So as if the sulfur is not a chiral, but if you can induce the enantioselectivity on that sulfur in order to get a better efficacy on that, a new proton pump inhibitor will be there, and gastroesophageal reflux disease can be treated like in this way. So that particular stereochemistry can alter the function of this drug in a very simple way. Okay? So now this information you should have, and you should have a proper tool in order to achieve the stereochemistry in the final products. Now to do for another very well-known examples of L-DOPA, that is actually the Parkinson disease, is applied for the Parkinson disease. But it has been found that the D-DOPA, that is not biologically active. So if you give it in a racemic form, that might create a different kind of troubles, or there is no use to give some more molecules to the biological target, there is no use. So you should have a method in order to make the compounds as a single enantiomer. So in these directions, the Nobel Prize is also awarded to understand this particular type of things against the Parkinson disease. You should have a enantioselective synthesis. The asymmetric thing should be induced properly in the synthetic route to get a single enantiomer of the drug, and that should be administered, not a mixture of like the enantiomer or in a racemic mixture. So these sort of case studies are hugely important, and accordingly the things are like people have worked on it, and huge contribution from their side to induce the chirality to get the final product as a single enantiomer. Okay, now to move forward, again going back, like coming back to that diversity oriented synthesis in biology as well as the medicines. They are the things like it is different, as I said, but before that we already discussed in the like other sessions that the high throughput screening, you need to have a screening of the molecule which is actually going to be your drug in future. So those compounds should be synthesized in a diversified technique to reach over there. So as I said, this way you can actually create the diversity. You can have different kind of functional group depending upon their polarity. They can combine in different permutation combination kind of arrangement to get a wide variety of compounds. Now this is the some kind of building blocks you can see, like here the molecules, they are having R2, R1, R3 and R4 part. Now if you vary different kind of parts, now you see in these particular slides, therefrom you can get few million of compounds by doing the permutation combination of the R1 to R4. Okay, so that way you have devised your synthetic scheme in such a way that you will be actually creating the library of compounds, not a single compound. So this way it is very important, and here also the functional group diversity will be actually making the skeletal, like skeletal in a diverse and very complex kind of molecules will be synthesized by applying the diversity. So the approach is changing. Where you can create the diversity, you have to start from there. You have to select that particular synthetic tool to get a library of compound rather to a single compound. Now how come it will be working? Just to give you some idea that how it is worked. So you see that it is a split pool kind of synthesis. You are having the substrate ready. You can see this is just some kind of cartoon image. You are having 3 different pot or 3 different container where you just keep, this is a video actually I converted in that one, so maybe it blurred. So you take the 3 molecules you are putting, all the molecules are same in 3 different container, which is having a tag, like it's a blue color tag in it. And then what you are doing, you are putting 3 different kind of reagents into it, which are like these colors, and allow them to interact with those earlier substrates. Okay, now what is going to happen, but in a different pots, different reagents you are putting. So now what will happen, it will be interacting like that. Initially the substrate was same, it is interacting with 3 different reagents or maybe another molecule or something. So that interactions will be same in like same means different in 3 different pots, 3 different containers. Okay, now what you do, this is the mixing of them. Then again you split them. So initially everything was same, you give them in 3 different containers, you put 3 different reagents or substrate into it, and again you split them, and then what you do, put it on a same container. So you mix them. Now what you do, you again, so now all the things, whatever the permutation combination are there, that is in a single container. Now again you split them, but how to split? Where all the permuted things will be there, all combinations will be there in 3 different things. Are you understanding that? So you are having 3 reagents or 3 different kind of molecules are actually interacting with it, and then again these sort of things will be same. These are all same, right? But you have already permuted them in 3 different ways. So now again you put another 3 reagents into it, and then again do this sort of computing, and splitting and pulling them in the same. Now you can see easily you can achieve this many number of molecules by changing the sequential use of the reagents from a same substrate. So initially it's a diverging approach. You are starting from one substrate, you are sequentially adding 3 different reagents at one stage. Later on you are again splitting and again another 3 reagents you are putting. At the end the whole scenario it is getting diverged. It's a divergent, you can get this many number of compounds in 2 simple steps. So this way you can get a compound like that, and of course like see these 4 different cases you can create the diversity in a single molecules. Your actual building blocks, the motif is same, but you can create the diversity by changing the sequential split pull approach to reach a library of compound in this way. Now how to go for it, the process, this process takes to the drug discovery, it's hugely lengthy process, we already talked about it, and cost involved, the expenditure is also very huge. So accordingly you have to have proper understanding and proper plan before that. There will be screening of it, then the synthetic, then the medicinal chemistry things will be there. What is your actual target? But accordingly you have to develop the process. So this is basically the parallel synthesis and doing different split pulling approach. At the end you should have the technique where from you can get a million of compounds in a very simple things, but at the end this sort of technique should be delivered to the industry, then only the scale up, the manufacturing things will be going on. So accordingly this I already talked about it, there is a space of those syntheses, diversity library has to be synthesized and correspondingly your products will be distributed. So this is also there, the combinatorial synthesis. Now here you can see if you take that A23 and B123, accordingly you can do all sort of combinations to get a basket of compounds with that. Okay, so that is also another way to get a library, there is a combinatorial synthesis. Then you have to go for in vitro, in vivo approach according to the what are the compounds you are getting, and you can move forwards for their industrial process development. These are the mixed split synthesis. Here also you can get 3 different baskets. Initially you get the 3 different baskets that is exponentially increasing. After that if you do another permutations you will be getting another. So 3 to the power of 3 you will be getting this many baskets, and accordingly that the number of compounds will be increasing if you move on with the process. Now the same way the combinatorial chemistry is also very important, where the different kind of things will be working like in a diverse way. So first is the diversity oriented synthesis, where you should have a tool by create the diversity, and now the combinatorial chemistry will be there to have the, you can see the targeted combinatorial arrays, the therapeutic target will be leading towards the lead discovery followed by lead optimization, develop the candidates, what are the candidates will be actually the drug, and then moving towards the industry for their large scale synthesis. These are all variety, but here the thing is you are just changing the different reagents from a substrate, and it might be some part it is a linear, and then all sort of permutation combinations things will be getting the library of compounds. So it is exponentially like increasing in that molecular entities generated by the combinatorial synthesis in these fashions, and you can see that power is on that. So it is not a linear, it is not an arithmetic progression, but geometric progression way the number of molecules will be increasing. Making the library, finding the active compounds, screening the mixture for their biological activities, and then you must have the proper synthetic tool in order to get the compound in a very efficient ways. And that should be involved, definitely the cost, because industry will be working for their profit and the other things, and also the mass manufacturing is important. Otherwise the product will be huge costly. So you should have a very, very smart synthetic tool starting from a easily available substrate in order to achieve the products, achieve not only a single product, huge variety of product, then only you can choose maybe few number of molecules which will be the drug. So it is a random screening or directed screening, you can have their proper structure activity relationships, but initially you don't have any kind of idea that which particular compound will actually be the optimization of that lead. So accordingly you have to devise your synthetic tools to that particular directions. So now that's what it can be coming, the discussion started from the natural products, because you'll be easily getting the product. Natural product you are not actually synthesizing at the beginning, you are isolating them and testing the biological activities. Then you are moving from the synthesis, followed by different sort of high throughput screening, keeping the natural product structure intact. You can create the diversity on it and go for their synthesis, create the library, and again go for their biological activity to optimize the lead. If you have a lot many options available, then only you can get that particular optimized products. Optimized means optimized in terms of their biological activities, and then only that compound will be converted, will be termed as a new drug. So accordingly it goes via different modes. It might be a oligomeric, different kind of peptide synthesis, some kind of template based combinatorial things will also be moving. This is some sort of library where people have already synthesized in it. This is a Schulze purine library. So keeping the purine intact, there are different parallel syntheses are done so that you can get a purine containing huge number of molecules by creating the diversity, and here from also you can get the product. That's a one bit one compound encoded library. Now you see, with that particular library you can choose that this compound you need actually for that corresponding biological target. So accordingly you can create the library. Here also, that is the quinoline library. You can see the diversomer reactions tube. Now you can have the synthetic route in such a way, this synthetic scheme I am not going to discuss, that way you can create the library, and therefrom one lead like molecules will be optimized for a future drug. Now you see that this particular compound is started like the from here, at the end there is R1, R2, R3 is written, but which way the diversity will be making. This is a library of that quinoline or this ciprofloxacin based the analog library, template based library. You keep that particular heterocyclic moiety intact, vary all the functional group, all the connectivity, and get the lead optimizations with that. This is also another number, total 480 member, the pool compounds. This is the proline based, it's a Gallup's mercaptil, like the proline based library. You keep the proline intact and do all sort of diversity in it. Now here also you can see like this particular compound which I talked about yesterday, that captopril compound. It is about 3 times more potent than the captopril. So that's what it is developing. So initially you have the drugs, but once you can do the diversity, you can get a lead optimizations which might be few-fold efficient as compared to your earlier drugs. So this way it's move on. Then you need to know that how these combinatorial things will be actually done, like what is the instrument and how you can do that. It is not the operations in a single flask and at the end of the day you'll be getting one product. Rather you have to make a instrument where different libraries will be synthesized in a very single time, like the same time all the reactions will be going on. You have to plan like a huge number of reactions by varying the reagent, by varying different substrate, but so that in a small time you can achieve a huge number of molecules. So these are basically the techniques, how it works. These are combinatorial chemistry equipments. Here you can see all the bottles are there, it's all attached. You can just put the substrate accordingly. Reactions will be going on. Even sometimes the synthetic robotic instrument is also there in order to get a huge number of molecules in a very short period of time. These also like that. Now high throughput screening already it's discussed. Some sort of things I'll be talking about. It is narrow down. It's a top to bottom. You are just approaching towards the bottom, and initially you have a huge number of molecules. You are narrowing the thing so that at the end you'll be getting only few representative molecules. Therefrom you have to do the lead optimizations for that particular biological targets. So accordingly this screening goes. These are different steps of it. Modern drug designing. So initially you can apply your designing part. You can start you can start your screening from natural products, from another drug also you can start. So in order to know that how the screening process will be done, what will be the filter on that screening, so that we apply and come down to a very few molecules which will be actually synthesizing physically. And then there are further biological studies to get the best compound among the libraries. These are some sort of different, like the fingerprinting region, which kind of substituents will be actually putting to cater or to enhance the better biological activities for that. And then there is some lipinuscule, I think, that is there, the oral drug properties of different kinds of parameter which will be actually associated to that in order to get some sort of ideas that which molecules might be having more biological activities. Now here you can see, like, this is this molecule, actually the small molecules which is coming from high throughput screening, and you do further derivatization of it, and see there are lot many other 460 compounds you can get from there. Then another compound developing candidate which is causing, like, against the anemia. But these kind of things I was just talking about, there is no chiral center involved. So you are keeping your diversity on particular directions just to skeletal diversity. You are attaching different functional group, different moiety into the molecule in order to like get lot many libraries in terms of their efficiency, like enhanced efficiency with that particular biological target. Now click chemistry is also very important, like the part of discussions here, because by click chemistry also you can create the diversity, where the two different functionality will be coming here, and just you do some click reactions, the final product will be coming in the chemical biology, new drug development. So that has been operated in the biological systems, and as well as the very easily here the proper functionality will be conjugating the antibody drug conjugates for the cancer therapy. These sort of things are there. The bioorthogonal chemistry is also playing the role over here. So you can just devise your synthesis in a click, so that in the presence of catalyst or in the biological systems, you can do such kind of reactions in order to get the better drug delivery as well as the efficiency of the drug. Now one question, like once you are giving the drug, you are administering to that, it has to reach to that particular receptor, but that might not be. There are lot many parameters which is already discussed. There solubility, so many parameters are there. But if you can have a synthetic tool where you know the molecule is already been devised, you just you administer the substrate, by in a single step that molecule will be synthesized, that small molecule will be done, so that it will be directly like reaching to that particular biological target. So it is somewhat the delivery aspects are coming into the picture in order to do it in a bioorthogonal mode. Okay, so this particular way, it's a orthogonal to each other. The chemistry is one part, you know the mechanism of the click chemistry and it is orthogonal to the biology. But if these substrates are administered on that particular target, the delivery aspects of the drug will be very easy. So in these directions, the things has been devised properly. You see that this is the drug candidates and this is the FDA approved things. Now if you do those kind of like pharmacophore together, and the efficacy of that particular drug will be enhanced. So that particular concept also coming into the picture, and most importantly here also you can create the diversity. You can just vary the substrate, you can give different kinds of group attached with that particular functionality. Accordingly that compounds will be like synthesizing in situ. So that's the way another Nobel Prize is also given. Like another Nobel Prize means it's given like for that particular ways, click chemistry with that bioorthogonal chemistry. And then you can see that again that Professor Sharpless is awarded the Nobel Prize with the Morten Meldal for their discussions on the click chemistry, followed by the Caroline Betajo, like the click chemistry to a new dimension that's inside the cell. It has been awarded, I think, like 2022. Now I'll be talking about some methods where the synthetic scheme or synthetic routes will be more efficient. So it is not about the like making the things in a glassware and you are just putting it. So that will be time consuming. That might need more reagents to it. So you have to make the method in such a way that continuously the products will be forming. Okay, so that's what it is called the flow chemistry. It is not batch synthesis. You are just doing the reaction in a single pot. That's called a batch synthesis. You do the reactions, then you take out from there, again you put another things. This is taking long time. Rather, you have to do it with a flow, with a continuous flow of the supply of the substrate in order to get the product continuously. The supply will not be stopping. That's what it is termed as continuous flow methods. So how it works, it's definitely they should be coming into the very quick contacts to it, and you have to keep the substrate and in a very the different channeling ways, so that the reaction rate will be faster. And accordingly you can find out that the continuous flow system, it is conventionally taking in a reactor zone. So different channels will be there to start from the substrate, which will be mixing to a particular zone, and the reactions will be taking place. If there is some other product or byproduct or the solvent, they'll be coming to another direction. Products will be coming to the different channel, and this particular flow methods will be continuously moving. So accordingly, the batch process and the different like the continuous flow method should be developed. Now the question is the process development. It's saying is very easy, but to develop this method is not an easy task. So that we'll be doing in some mode in this way. You can have the things in two different channels which will be going inside the reactor. You can have like to have a facility to change the parameters. You can vary the pressure of the reagents. You can vary the temperature. You can just change the size of that one and how fast it will be going. You can apply some external force also, maybe the microwave, maybe heating, or if it needs some other kind of energy source, light you can give, and at the end the product will be coming, purifying and collecting in the other end. So accordingly, this particular type of techniques developed. Of course, that will be very much advantageous in terms of not to have a purification part, not to have a maybe less amount of solvent, reagent will also be less. You can accelerate the reactions, you can apply some other external like pressure or some kind of parameter on that in order to get the product in limited time. You can have reduced the cost of it, but devising of this particular type of things are not an easy task. So accordingly, the things are formed like different cartridge. These sort of things are used. There is a company who are actually doing on hydrogenation reactions. Fortunately, I have used that thing. It is like a cartridge you are taking where the catalyst is embedded. Like inside that cartridge, the catalyst means for hydrogenation, it is like palladium, platinum based catalyst, nickel catalyst. So inside that cartridge, the catalyst is already there. You put the substrate from one side and keep the hydrogen on it. Automatically that hydrogenation reactions will be going on and it will be coming to the other side. You need not to do, you have to only take a pump to make the flow of the reactions. Once the pump is there, it will be pulling, it will be sucking the things from this side and going in the other. If you see the reaction doesn't go, then you can channelize them again through that cartridge. So that's what the in-between, that is a reagent systems where the reagents are installed, reagents are embedded on that cartridge through which the reactions will be taking place, and continuously the product formations will be happening. And rather you can regenerate that cartridge, you can vary it, you can change another cartridge, you can change so many different kind of catalyst into it. So you need not to do actually the lab work over there, but you just guide the, and most importantly, these are all automated method. You need not to be steer, you can just automate it. N number of reactions will be one by one going on. So types of flow reactors, there are different gas, like the column reactor, gas reactor, depending upon the reagents, depending upon which kind of reactions you are actually going to do, and accordingly you can vary all sort of parameters here. These are the there are a lot many companies are actually developing these sort of things, and development, as I said, it is not an easy task to target something, but industrial scale synthesis might not be like, maybe a difficult task, because for industry you need to get the molecule in huge amount, not a small one. So in terms of making the cartridge, in terms of making the flow in this level, that is something a task, huge task. But beginning where the high throughput screening or library synthesis is happening, for that this is very important instrument. You can keep it together so very quickly, or like without the lot many solvent expend, or maybe the different reagents, you can get huge number of molecules in a very quick time. But the limitation, of course, as I said, there might be clogging, maybe the flow is getting blocked, then you have to again operate it, open it. These are the difficulties to handle it in the industry. So those aspects also should be taken, and it's environmentally, like the future challenges, definitely environmentally pure compound should be there, less solvent should be used, accordingly the things will be going. These are like the continuous flow synthesis of ibuprofen. You can see here that ibuprofen, that is a non-steroidal anti-inflammatory agent, right? That ibuprofen, you can get it from any medical store. It's called brufen. Okay, now you can make it in this mode. You take this particular type of substrates and see that continuously with the column, these things are going on, but that is outside, but inside you will be having a complete instrument. You will be having some channel. You get the substrate over it, vary the conditions, reactions. At the end, the other channel, you will be getting the product in this mode. You can see the picture in these particular directions. These drugs has been synthesized by using the continuous flow method. But developing that method itself is a big challenge. You cannot, maybe it is possible to achieve different kind of drugs, that is not there. Like it's a challenging part. Maybe tomorrow that challenge will be solved or that things will be working. This is also continuous flow method for the synthesis of that provitamin D3 along with this particular continuous flow method. So there are the research groups who are developing the method, like they are not doing any new drug synthesis, but if the synthesis is developing through a continuous flow method, that is also a very good contributions on that domain. Okay, so this way you can make the generalizations. Maybe later on that, you know, what are the reactions we are doing in a glass apparatus, that any conical, those things will be obsolete after a few years. So always if you can make something, it will be having a device, you attach them and getting the product. You need not to do any kind of purifications, nothing will be needed in between. But development of the method is a big challenge. These are also the, so like you can see how the tamoxifen have been synthesized. So it's a 12 gram, it's prepared, it's about like 84% of yield. It's simple starting material are pushing on into that particular cabinet. After certain stages, different kind of conditions have been given, and you will be getting the final product in a very pure form. Now the thing is, it is continuously going on. If it is not something problem, again you can start from the beginning. So this way you can devise the things and you can make your synthesis process or the that process will be more accelerated. Now I'll talk about some another aspects that is called DNA encoded. How much time? No? Okay, so we have time. So DNA encoded chemical libraries. Here what is important is the phase display. So you can see the DNA, there's an organ, like it will be phenotypes or genotypes. This way you are just attach our DNA and there will be some kind of recognition of the small molecules with the DNA, and you can move on in that directions. This is also a library creations where the DNA will be the part of it of the small molecules, and you can create a huge number of libraries by using the DNA as a tag on the library. That's what it is called the barcoding. It is encoding. You are just basically taking the DNA as a tag on the molecules and you can create the library. So this way it works. It's the collections of organic compounds individually coupled to oligonucleotides or DNA fragments. So the compound should be actually coupled with the DNA in order to give a tag to it, and accordingly the fragments is serving as an amplifiable identification barcode which will be tagged, which will be coded, and the embodiment of a phenotypes is a small organic molecules binder to a target protein. That is actually important. So this is a concept of creating the libraries where DNA will be using as a tag, and in order to move forward that barcoding things will be there. Now it might be a genotype things, a DNA sequence that permits the identifications of the corresponding phenotype molecules. So DNA actually coming from the genes. So the phenotype things will be there with the small molecules, and that combinations of the small molecules will be with the DNA will be there in order to give that barcoding things on the library. Accordingly, you can have the phase display, yeast display, mRNA display, ribosome display, or selects. So this way the DCL, the technology is originally proposed by Lardon and Bernard and allow the construction of peptide libraries. So peptide synthesis is also a big task, the protein synthesis and all. I'll be talking another aspects in it. So that DNA will be attaching with the small molecules to give you the tag of it and barcoding those libraries. You can have the once you are putting the libraries, you can give them a tag that this is, the barcode, how it works. You can put the laser, immediately you'll be getting the tag or which kind of product it is. So these sort of directions you can use by using DNA as that tag. Accordingly, you can get the corresponding libraries. So how it works? It is the represent a versatile and powerful technology in that platform to the discovery of small molecule ligands with a protein target of the biological and the pharmaceutical importance. So that's what the tagging will be important. Once that small molecule tagged with DNA will be reaching to the particular biological target. Accordingly, you can get the different kinds of property changes and all. And now you can see that DELs are like collections of molecules which are independently coupled with DNA, and that tags amplifiable identifications of the barcode. Now, once the DNA is present, you need to see that you are having a basket of molecules where every molecules are there, small molecules tagged with DNA. You put a laser kind of things, immediately you will be getting this molecule is this molecule, all the information will be coming. That's what is the barcoding. The identification of things will be easier once you are tagging your small molecules with the DNA, and that one will be recognized by the protein target, whatever the corresponding receptor you are adding. So this way it goes in this mode, and at the end of the day, that the validations of that particular type of like screening, and at the same time the sequencing and decoding will also be there. So at the end you just decode the DNA, you just identify the DNA, you couple the compounds with DNA, you move towards the protein target, and at the end you just cut off the DNA. That is very easy to get rid of the DNA, but that barcoding things will be there, and you can have a huge number of like DNA encoded libraries could be synthesized. So that is another approach to have, and this way the huge number of drugs discovery work is going on, enhance the lead hit identifications and the lead optimizations. So once you are just coupling the small molecules with that, so we have gone through the all process, like where from it is started, that computational analysis followed by the high throughput screening and all sort of different kind of like filtering. Now the AI also part of it. But after that, once you are having the molecules, if you can tag them with the DNA, that can be used as a barcode of the compound. And at the end, once it is moving on towards the protein target and this, then you can decode it again. You can just cut the DNA out from there, and that molecules will be delivered, that molecule will be reaching to that particular receptor. So this way it works, and DNA barcoding enables the accurate tracking of biological molecules. So where it is actually going, the molecule, that also could be properly tracked, because DNA, that particular tagging will be moving along with that one. And DNA, it is a biomolecule, so it will not be creating any other, the side effects or any other difficulties where moving to the biological systems. So that way the biomolecules will also be very important, like the useful in these particular directions, and the barcoding things will be going on. It will be very much cost effective and scalable for the pharmaceutical industries, because DNA you can extract from different plant sources or anywhere, and accordingly that coding, because why DNA? DNA is a unique thing. Okay, the DNA might be different, so you can have a proper recognition of the small molecules with that particular DNA, and once you are administrating the things in the biological systems, that flow will be there, that compounds will be moving along with that biological systems to reach to the target, and every aspect you can track them properly. So this way the DNA encoded libraries are very, very important. It is used for the high throughput screening, for the drug target identification. It also supports the advancement in the precisional medicines. So once you can tag the DNA with your small molecules, and then the precision that will be target, the drug target identifications will be very important. So you can see that where the binding is happening. I'll get some water. So the small molecules, if you leave it, it might interact with multiple protein targets, but once it is tagged with the DNA, then you can easily track that where are the targets that molecule is targeting or interacting with which kind of amino acid inside the protein. So this tagging is important here in order to understand. Now you can vary both the things. You can vary many small molecules where the synthetic things already we have discussed in the diversity oriented synthesis. After that, if you can tag them, so one sort of library you can synthesize by applying your the sequential synthetic tools, then you attach the molecules with the DNA. So initially it was a chemical library of the all chemical compounds, small molecules library, combinatorial libraries. After that, second steps you do, attach the DNA to all the compounds to get the DNA encoded libraries. Okay. Now that particular way you can have an attachment will be possible to that, and then these small molecules you can put study the interactions with the protein target, and in every cases you can properly track them. You can understand their delivery aspects. You can understand their interactions with different kind of amino acid residue in the protein. So this way these libraries will be very important for the after the high throughput screening. And how it works, it's like basically anchors with this one. So the structure of DNA is known. If you can anchor them with a group which will be functionalized with the DNA. Maybe here that is there as an imine, like the dynamic assembly with that one, and then the heat identifications flew with that one, and then at the end you can just reduce it. The reductions will be there. So initially, if you can look like the like see their chemistry, initially you are having a tagged aldehyde which is actually interacting to get the CIP base, corresponding CIP base with the amine to get the imine. Basically it is imine forming. Now that is moving on to the target binding, and at the end if you can reduce it, you will be getting the corresponding amine. So which way the imine will be interacting with the protein targets and which way after the reductions the amine will be interacting, that very easily you can basically monitor or you can track through the tagging with the DNA, because DNA part will be intact. The long chain, that particular part will be intact over there, and that is not going to create any additional like disturbance or additional problem or maybe side effects when it is moving or interacting with the biological things. Now after that, what do? You can have that capture of the primary amine, not the secondary amine, and sequence the elutant. Okay, so you can get rid of the things very easily. So you can get the primary amine. So because the primary amine will be there from the DNA, you can get rid of the DNA and then what are the compounds are here, how it is actually reacting, you will be easily knowing. The heat identifications will be very easy. So like once it is started the medicinal chemistry discussion, it is the identification of the heat and optimization of the lead. These two things are you need to keep in your mind. But how to identify that which molecule is actually interacting with the proteins, that will be very much easy if you code them or if you attach the DNA part with your small molecules. So that's what the DNA encoded libraries are hugely important after the combinatorial libraries, so that every molecules will be tracked inside the like biological systems in their in vitro studies. Okay, so this way it goes. And at the end it is very easy to decode it. You can just get rid of the DNA at the end to know exactly the interactions and how it will be going on. And you can get rid of the interaction, because since the normal amine versus primary amine versus secondary amine or the imine versus amine things will be operating, it's very easy to identify their corresponding interactions with the protein target. Accordingly, the the monitoring of the small molecules or whatever that the knowing the heat identifications will be very much easy after this kind of screening. Now to move on, what happened? This I talked about. So how it goes? So look at it. The magnetic beads is there, and then followed by the resin. That resin tips will be like attached with that, and interaction that dependent on that, and followed by the photo affinity labeling. So you have to label them and to move on the corresponding libraries. So this way, what are the chemical libraries were there, you can transform them to the DNA encoded libraries. So this way the process will be going on, and accordingly a lot number of molecules like this. So derived small molecules ligands, and that is a macrocyclic ligand. So all these sort of things have been actually coming from the DNA encoded libraries. So huge number of molecules is actually interacting, but of course there are limitations. You might not be having the appropriate functional group. Barcoding things are, it is one way to move on, but it is not maybe possible with that, or else you have to create that functionality in your small molecules in order to attach them with the DNA, and then followed by you can monitor or you can track them in that particular encoded like the libraries with the biological target. So these are basically the limitations, but still you can see that how different kind of small molecules are actually coming there, and all sort of cases there are biological activities, there are the KD values, there even the ICP value in some cases, that will be actually coming good in terms of understanding the which is the best heat on that, to identification of the heat in this particular direction. Now I'll talk about another approach which is also very important in a continuous flow method on kind of things. It is a microfluidic techniques. So it is basically the revolution in the high throughput screening and the precision medicines. Microfluidic means the amount of the things will be very, very less. It's a micro droplets. You can apply, you can put their drugs on it, and to understand their corresponding like the identifications of which kind of molecules in another kind of like continuous or a automated mode. So here you can see the microfluidics that involves the manipulations of the small fluid volumes in the micro scale. So you have to take the things in a micrometer, that particular dimensions, and accordingly you can get different kind of, you know, the pellets or different kind of spots on it. Enables for the rapid chemical reactions, precise control and automations in the drug research. So you can have the plates over here, you can have the droplets of the molecules that compound in a very small micrometer region. Okay, accordingly you can get the fluids, but it should be a continuously moving. It's a fluidic things will be there. And that integrate the chemistry, that all the things, the engineering, the efficient drug screening process will be very much important with the technique. So this is the process development. You can have the like the small molecules in a form of a spot, in a form of a micrometer region spot, in order to get a plate of it to understand that how the that particular things are operating on it. So key features is the miniaturization. If you are just narrow down, you are making the things in a very small. Earlier your concentration is important, how much concentration you will be putting, accordingly the in vitro test will be undergoing. But here what you are doing, that reduces the reagent consumption and the waste. So what are the things you are doing, that you are doing in a spot on the microfluidic ways. So it goes for the high throughput screening, it rapid analysis, it's a plate, you can give a like the lot many analysis will be done in a very quick approach, and then the thousands of compounds will be actually screening in a very short period of time. It's a real-time monitoring, enables the life cell analysis and the drug interaction studies through this microfluidic approach. Okay, so this way the huge amount of work which is actually going on in a different mode, basically I would say the maybe in the in vitro part, in different modes, that can be done in an accelerated way by making them in a microfluidic channels. So the automations and integration, it's merge the synthesis as well with the screening and analysis in the one platform. So you can start with, you do the synthesis in a microfluidic way, whatever the output is coming, that you can do a spotting over there, and their biological activities will also be tested on the same platform. So this way that microfluidic systems will be operating, and that this particular type of process will be developed in a really smart way, or really like a very less time but huge efficient ways. So how it works? High throughput screening is a fast screening of the drug candidates using the micro droplets. So you can see, like you can put it even on the skin, on the channels and organ on a cheap models. So you can keep them over there, by which the thin film where you are putting the spot, that will be modified in this mode. So stimulates the human physiology or the accurate drug response testing, single cell analysis, the nanomedicine development. You can use the how small the medicines in the nanomolar level that can be developed through this particular type of techniques. Now this way that that can be correlated with the IoT, the Internet of Things. So once the things are in this mode, you can correlate the things. It will be using in different kind of sensing. What are the diagnostic tools are there? Here also you can use them. You can track, you can get the proper data out of it. What are the parameters are there? It is there on that small droplets, which will be actually doing for all sort of diagnostic tools. That similar way you can do the synthesis also in this mode. What are the continuous flow methods? And at the same time the test of these small molecules with the biological target, that also could be done in this particular mode. So that's what what are the channels are there in terms of have a identifications of the heat, that is the high throughput screening, followed by the optimization of the lead, like which is actually coming on that to get the best efficient drug, and their synthesis. These all the things could be made to bring out in a single platform. So that's the way the microfluidic technique will be hugely important in terms of drug discovery as well as the drug development approach, okay? So this way, this is very efficient, and moreover, it could be automated very easily. With that one, you can get, apply them for different. Nowadays, you can see the delivery approach, the drug delivery things are different. That painkiller, you used to take it orally, oral painkiller used to have, but now you can give a patch of it, right? Where the pain is, maybe in the back pain or where, you can put the patch of that one, it can directly deliver properly in that particular point of that one. So these sort of things are there, and moreover the use of that things are very less. It can be correlated to your smartphone as well with that device, that is the IoT. Maybe the electronics persons will be more than happy to explain those things. So whatever the data you are getting, that will be coding, that will be there, transferring to some kind of data bank. It will be immediately understanding, so that now in future generations of persons in having whatever the parameter you are having, they are like the blood parameters, there are different kinds of like the changes, it could be easily available in your smartphone, okay? So that's the technique there. They may be putting, there's a lab on a chip, you can have your all parameters of the human body and on just a chip. If you put it, immediately those informations will be coming from there, and that you can correlate in your smartphone. You can share the things to others. Maybe you need not to like go to the diagnostic place to do your blood test, these things all. You can just share your data to someone, they'll be understanding what are the like the conditions over there. So this way the microfluidic techniques actually sorted out to all the things which is needed for the drug discovery or development. But again, it is not a very easy things to just like that coming into the market. It will take huge amount of research on it. All the things may not be working in these microfluidic systems, so those optimizations are needed. Accordingly, you can devise, you can frame the corresponding techniques to get the droplets, the micro droplets, in order to do that particular type of delivery aspects. So this way, just a diagram, you can see that how colorimetric reagents will be changing. Like if you have that kits over there, that drop of blood you put, accordingly you see that how that colorimetric detections will be done. Accordingly, the things will be changing in that direction. So you can have the like their waterproof sticker paper, that the plasma separation membrane, the filter paper, and printed on that, you can keep those particular type of small molecules into it and put it on the skin. So accordingly, once it will be interacting with the bloods, with the color change, the colorimetric detections, the things will be operating. So it will be doing in the like the diagnostic as well as the therapeutic applications. Here also you can see the huge amount of work is done on that particular direction. It's a sweat biomarkers by the temperature. Sometimes the diagnostic things doesn't work, it doesn't need that always you have to go for the blood test and all. If you have the data from your different like biological samples, maybe sweat, maybe the other different kind of fluids, you can get the similar idea, but you should have a proper biomarker in order to identify them. You should have a proper molecules, you should have a proper developed chemistry into it, proper colorimetric detections in order to understand that thing. That is as well as for the therapy, as well as for the diagnosis also. So this way that all microfluidic channels and things are there, and all most importantly these all informations, it's a huge informations you can get. Even not only that, you can make it like enhanced to lot many different kinds of parameter you can very easily identify, and most importantly that can be correlated with the IoT systems to store the data, to transfer the data in some like some smart device or gadgets, okay? So this way the techniques are hugely important. Now another also important techniques, that is automated flow, like the peptide synthesizer. So peptide things are very important with the protein. So if you keep the amino acids and to attach them, you can enhance the like their chain length and all. That is very, very peptide synthesizers. There are a lot many drugs which are actually containing the peptides, the small molecules, amino acids. So how to connect them, how to synthesize them, that is also a very big task for it. You cannot just do, you know, you have learned the peptide synthesis maybe in the undergrad classes, that you can have some coupling agent, take one particular type of amino acids and then put the other one with that coupling agent. then that CN coupling will be happening. So this way you can move one side your either N terminal or C terminal, and the other side it will be propagating like that. It is nothing but a polymer, okay, the peptide, that is a condensation polymer, that reactions will be taking place. But for that you must have the proper coupling agent, you must have the solubility issue, there will be the separations, the purification things are hugely important. But in terms of doing all other things, you can have an automated peptide synthesizer. So what you can do here, you have to take the amino acids with a particular protections in the N. If you want to go for the N terminal things, you have to take that nitrogen part intact and you allow it. It's a completely automated instrument. Just to put that, this will be coupling with that, followed by the other one, that automatically the peptide will be synthesized. So one after another sequential form, the amino acid will be coupled to each other to give you the big peptide. Otherwise, synthesis in the lab, that is very tedious job and sometimes not very fruitful also. This peptide may break during the purification and other. But here you can just easily automate them. You can keep different channels of it. As you can see here, different channels are having different amino acids, and then through that particular column, it will be moving. You can vary their corresponding reaction conditions. You need not to apply lot many like different kind of coupling agents, but at the end you can get the peptide in that particular sequence. Sequence is very important, okay, that should not get interact with each other. So this way the automated flow, fast flow peptide synthesis that works, these sort of instruments are very, very important thing. And as I said, this sort of like the development is not only done by a chemist, maybe chemical engineers, maybe the electronics engineer, they are also involved here. You have to write a proper program in it. Computer science are also involved here, so that one things will be properly automated, and in order to get the corresponding products in a very fast and accelerated way and hugely efficient, that will not only cost your time, and most importantly, they are very much, the accurate. If you want to do the things in the lab or manually, first of all, it is not very viable. At the same time, you might not be having the proper selectivity. That might be mixing. If this amino acid will be coupled to other, at the end of the day, there might be interactions or the interference with different things. So multiple product will be forming. But here the things are very well automated. The things will be coming one after another and coupled to give you the end product. So this way the techniques are very, very important. Advantages is the faster drug discovery, the real-time analysis, lower cost, of course, like due to the minimal reagent use, the operational cost is of course lower, but now capital expenditure, that is not lower. But today's peptide synthesizer is huge costly. But maybe after a few years or so, the research are going on, so that might be very much easy in terms of making the things automated. Then of course the lower cost due to that higher precision and reproducibility. It is hugely precise, it's hugely accurate things will be getting. That is the reason it is costly. So you can have all different amino acid in different channels, and accordingly you can have the program that this amino acid 1 will be coupled with maybe 4, 3 will be coupled with that, then this particular coupled product will be coupled with one more coupling. Are you understanding? It is not always the one amino acid will be coupled with 1. 1 plus 1 will be happening. Whatever that A plus B will be giving you C. Now this C will be coupled with D, or maybe D plus E. Then in this way you can increase the chain length of that peptide. Okay, so this way the peptide synthesizer work, and it is a automated like the flow control technique by which you can get a huge amount of big amino acids like the proteins, the peptide in a very easy way and less consumption cost. I think these things I already talked about. So how much is that done? I think so. Now I think I can stop here. This is almost all the techniques we have discussed about this particular process development. So you can ask some questions in that directions. I have many other things to say, but I think it is time also approaching, so you can talk about it, and then we can discuss the other things. So please go for some questions.
Hello, sir.
Yeah.
In DNA barcoding, what it means telex?
Okay, so these are basically the different techniques. Okay, now once you are keeping the DNA, which particular molecules will be there attached with the DNA, that is not fixed yet. Are you understanding? So all the molecules may not be attached with the DNA. So that's a screening that this molecule along with the functionality will be attached. In this way it goes. So this particular way the library may not be created. So that depends on what is the functionality is present on that. Accordingly that library will be created.
What it works, selects means what and what it works.
Exactly the meaning of the term I don't know, but that works with the functionality of the small molecules. So as you can see there that you have to keep the functional group, because you need to understand DNA means what kind of functional group. You can get the amine, but in your small molecules there is no other functionality which will be attached with the amine, then it doesn't work. So that coupling is important. Now that's what I'm saying. You are having the chemical library, but not necessary all the functional group will be identifying by the DNA. That is not possible. So if you want to create the library, you have to take the small molecules, that whatever the initial combinatorial library you are giving, that should be attached with the DNA with that amine of the DNA. Or if you want to move on, then you can create the aldehyde also over there. Maybe in your compound you are having a hydroxyl, let's say one alcohol group is there. So just to oxidize it to aldehyde in order to couple with the DNA. Okay, or maybe you can get some acid also to get the peptide coupling with the DNA. So this way the idea is that you have to attach the DNA with your molecule without disturbing the normal function of it. And then you can tag it. So once it will be moving, you can tag the DNA up to the reaching over there. That's a tracking things. Okay?
Thank
So
you.
yes.
Anyone?
Any other questions? Yeah. I hope you understood the things, right? In a sequential stepwise. Yes, ma'am.
Sir, is there any tool, web tool for checking the synthetic feasibility of compound? Because we perform the synthesis by conventional method.
Okay.
In that, sometimes we get the product or sometimes not. And in this process, most of the time the chemicals wastage occurs. So
Yes.
that to avoid this, can we check the synthetic feasibility?
I think there are some kind of tools are there, but you know that is not very much reliable. That how much you can rely on it. There are a lot many AI related things are there. What algorithm that if you frame the synthesis in this mode, which way you can devise or you can frame your synthetic route. But that might be that is depending on the accuracy of that algorithm. So you can just test it. They are also doing the similar kind of thing, but it may not be 100% accurate. So you can get some idea that in these directions maybe it will be going good, but it may not be completely like, you know, and if it is 1% also less, that might be affecting the actual your experiment. Okay, so better you need to have a balanced way. You can apply your chemistry knowledge over there. Otherwise, you need to understand after a few days, if you see all the things are automated and this one, then that chemistry knowledge will not be implemented properly. Now, if I say, okay, you are having a secondary amine, that whatever he was talking about the pK value, all the things, you can check it very easily by doing the titration. But if you have that algorithm, that might not be knowing that what is the other effect will be operating there. Are you understanding? If you have the normal, let's say reductions of your aldehyde or maybe nitrile or this kind of group. So you can keep it in the algorithm. You will say, okay, for these reductions you have to use, these are the reducing agent. Maybe sodium borohydride, a very simple reducing agent. But that algorithm may not be very much efficient to say if your cyanide is there, nitrile is there and another ester is there, which one will be reduced. So the things are very diverse. Now applying our chemistry knowledge, we can say, okay, if you can use the sodium borohydride, maybe this particular group will be reducing. Or if you use sodium cyanoborohydride, maybe the other group is reducing. Or maybe in some cases you need maybe strong reducing agent. So these are, but the algorithm doesn't have these things to exactly predict that what is going on. But you can definitely check it out. You can definitely have some kind of, you know, the ideas that which directions it will be. That's what people are working. Like I know like lot many organic chemists during last 5 to 6 years or maybe maximum by 10 years, they are publishing huge number of papers in the AI generated synthetic techniques or AI in organic chemistry in order to get the proper screening, that what is your actual question, that what will be the best synthetic techniques to reach the molecules, or maybe what is the best synthetic techniques to induce the chirality on the molecules. This sort of algorithm people are working. That's really a big challenging task on that. But once as a synthetic chemist, means once you will be adopting it, it might not be completely depending upon that, because your molecule may be different as compared to where the algorithm is generating. Because that particular thing, it's a feeding data. You have to feed the data into it, that okay, this many number of cyanide you are feeding as an input, that maybe the benzonitrile or like benzyl nitrile, you feed it, and then accordingly what are the reduction are happening, that data you feed to the algorithm, and then accordingly it will be predicting. Right. But if I say you are having benzonitrile attached with an ester or a nitro or another reducing group, it might not be accurate. So that's what it is not up to that reliability. That is accurate. That is also a big contributions that it can predict, but it may not be that much practically accurate in that domain. So,
Okay, sir. Thank you.
So these discussions, the timeline is also very important, that whatever I talked about the target oriented synthesis, the retrosynthetic analysis, that time the objective was to synthesize the molecules. So you have to be devised enough to get that compound what you are targeting. But afterwards, that particular targeted synthesized compounds are not enough to cater the biological importance or the biological necessary. Okay, you are understanding, okay, these molecules will be having anti-cancer property like Taxol and all. But maybe Taxol is not good enough to cure that job. So you need to have an alternate of Taxol, but which alternate of Taxol will be synthesizing? So then you should have a library, you should have a technique of that, where a bucket of Taxol kind of derivative will be there. Now you test it again, that one. So that synthetic technique should be ready before approaching to that disease. So that way it moves. So this way the diversity things, all these combinatorial things will be coming into the picture. Later on the automated things are coming. So the timeline is very, very important into it. Earlier I can understand, I tell to my students also, many, whoever are our teacher, many of them are worked on the natural product isolations, they are characterized. That's it. That is also, that time it was hugely important work, that you can isolate a natural product from that particular tree, and the structure is that. But today's date, if you do this research, nobody will fund you. Because why to identify that? Unless, if it is really having some huge medicinal property, then only that is there. Otherwise, if you directly go for the natural products and isolate them and do the synthesis, make a hugely big, and you know that all these sort of chemistries are, it's not a very easy, it's a costly, this sort of things are huge costly. You have to develop different reagents, solvents, all the things are involved. Now if you can reach also with that particular targeted compounds, what will be the benefit? So these things are involved now. Okay, so this way the time-wise, timely basis, the objectives, the development things are changing. Yes, any other questions? Yes, please. Yeah. No. Molecular docking is a theoretical concept. Molecular docking you are doing, theoretically you are just identifying, you are designing the compounds, and you see how your compounds, you are just leaving your compound in a protein environment, and you see how your compound is interacting with different things. Now if you see again, if you just go for the literature, you will see in early 2010 to 2015, in this particular time period, huge number of paper published in silico analysis. Molecular docking, you do only simple this kind of computational things, and you can get a very good paper. Even very good journal also you can publish a paper. But what is the reliability? They will say it's a huge binding. What is the parameter there? Binding energy, and to understand how it is interacting with the proteins, right? That's the things you are getting as an output. But is it actually working once you are administering that drug into it? Maybe no, not always, that particular reliability is very less. Even I don't know that how come once the the existing drug, it is beaten by one synthesized molecule, and whether that is actually coming into the market and like a so-called drug is getting approved, I don't know. That is the big challenge. These are all computational things, but DNA coding is practical. You are actually attaching the things with the DNA. But it is not like that that's what I said. The library you are creating in order to understand their medicinal application in a very concise ways. Okay, so now if that is not possible, then that's what I'm saying. It is not always possible that every molecule could be converted to the DNA encoded library. That may not be possible also. Yeah. So it is not, that's what the tech, but it is hugely important. If you can tag it, you can understand the things, you can track the molecule properly inside that, you can, whenever the molecule is reaching to that, those things you can very easily understand. What do we, okay. You mean whether that will be attached with DNA? Maybe I'm not on. No, I understand. You are talking about one protein that is attached with this one, and then you are understanding their interactions.
It is already in the protein library.
You
with
mean
the
the
existing molecule?
How it will
Like one is the what can I say? The MAO-B inhibitor, rasagiline.
Okay.
Right. There is a one protein like 2C65. It is in a PDB. ID is 2C65. But this protein includes the rasagiline structure also.
Understood. That crystal structure is already sorted out.
Yes.
Yeah, that's a
So
crystal
could
engineering
it be
thing.
also called as a gene encoding?
No. Where is the gene? There is no DNA.
Sorry. But there are some DNAs are also available in PDB.
No, understood. What you are meaning, that properly understood. See, PDB is a bank, protein data bank, right? It's a huge number of proteins where the structures are well defined. All are having the crystal structure also. Now, crystal structure of a protein will helping you to get that molecular docking things. You get the structure from there, you put your molecule on that protein structure is already known, and you can see all sort of interactions. That's what the in silico studies are done, right? Now, if you can crystallize the small molecule along with your protein, then that's the concrete result. That actually tomorrow, whatever he'll be talking only on crystal, that protein crystallography. So you need to understand, one is the crystal of the protein, another is the protein along with the small molecules. So whatever you are predicting that interactions will be there, maybe this much bond angle, this one, that one, you can actually see in the crystal structures that the small molecule is there and how it is interacting with that particular, the active enzyme site and this one, active site of the protein. So what you are talking about, you are having a crystal of the protein, you are having a crystal where protein as well as a small molecule is there. So it might be a DNA, this thing, but it is not a encoded kind of thing. That is not I'm saying. So this is a practical thing. You can have a small molecules. Whatever I am talking about, that is the synthesis of a huge number of molecules. Now, what is the objective to synthesize that many number of molecules? That is not known. There is a big question mark. Whether this will be helping or not, nobody knows. Maybe none of the compound is actually serving the job. That is there. Are you understanding? So initially the high throughput screening, theoretically you can say, okay, this many number of molecules will be coming, maybe crossing whatever the parameters are, solubility, that log P, these all the things whatever talking about, their PK values, their toxicity, advanced studies, all the things are done. But you are not synthesizing, you are doing it theoretically. Now after that, if you don't have the molecule in your hand, how will we optimize that, identify your hit molecules? So for that you need to have a tool in order to get the molecule in your hand. Now after synthesizing also it may not work. That's what it takes that much time to give a million of molecules to get maybe one or two compounds. Whatever you are saying, that is the understanding of the molecule in the presence of a protein, that the crystal structure of the molecules will be there, how it is actually interacting with the protein. That is another PDB you are saying, that is another kind of structure. But here it is a tagging, you are understanding the things in a practical ways. You are actually attaching that DNA along with your small molecules, and then you are putting, then you see that how the things are going on. It is, you are not at all actually knowing that how the interactions are changing. DNA may not be changing, or maybe the more efficacy, no, but it can track where it is going, how it is going, practical sense, that will be there. You can encode just exactly like you put the laser, you will see, okay, here the molecule is present. Okay, so that's what you are making that library.
Thank you, sir.
Yeah. Other question? Any other questions?
Study or
No, I think that is already in the market.
okay, it's in the market.
So I think what I'll be doing, I will share you at the end maybe few sources. There is a Chiralpedia. There is a actually who is the founder of that Chiralpedia. He's very close to me. I invited him also to like maybe online mode to deliver a lecture, maybe 1 hour. But he's a retired faculty, so he is having some prior this one, he could not. But that is a really important, like the blogs or all sort of things are there. It is like Wikipedia, you have understood. Now it is a Chiralpedia. Like all sort of chiral drugs is included there. That's a free, you can just easily search. I get from there only. I already speak before like getting the thing. He's very enthusiastic persons. Even like I don't know, maybe you can interact also with him. So here what the things are done is a lot many different kind of pharmacological informations. They are the drug development, all the things they are just making in a single site. That's what like he's the founder of that particular things. It's a Chiralpedia. You can maybe Google it also. So all sort of chiral drugs that is included there. There only it says the importance, why that thing is important. Now you need to understand at the beginning, once you are isolating the natural products, you don't know anything about the that stereochemical importance or this one. Even you don't know that whether it is actually having a chiral center or not, or whether you are isolating it as a mixture of enantiomer. That you don't, those idea you do not have. But once you are actually moving to that particular biological things and you are studying them in different ways, you need to then identify what is the role of that chiral center on that. And it is, you can see it is entirely changing their biological efficacy, their different kind of properties, their flavor, color, it is entirely changing because of one chiral center, just a orientation of the group. That's what the shaking hand thing is very, very important on that. Otherwise, it might create some different kinds of, the side effects, another kind of disease it can happen, or in some other cases, that is also good part, that it might, the other enantiomer might be good for other disease also. Are you understand? Not to talk always the bad part, maybe if you put the two enantiomer together, one will be working on that, other might be working to some other one. That's what the neprosen, whatever you are. So if you don't have the chirality, that R1, the sulfur, it is only the omeprazole. That's the liver, that stomach. That is indigestion. The medicine is there. But if you put it in a chiral way, that you can solve the other problem as well. Just that the high acidity, that acid reflux, that will be working better into that. That is a good part actually. Both the enantiomer is having different biological, and like if you have it in maybe combined way, the actual biological efficiency is enhanced. But for thalidomide, no, that is not. That thalidomide, that is a bad part. That is a huge tragedy. Okay, so this way you should have a complete understanding of the efficacy or the structured things with this one. Okay, so that's it.
Sir, from where we can avail the facility of high throughput screening technique?
I don't know. You mean this one? These are actually, that's a technique, but the facility means if the things are done, you mean you want to do the high throughput screening?
Yes, yes.
I don't know, maybe there are database, this one, but really I don't know about it. So high throughput screening, it's a method. You can screen the molecules to get this one. Then maybe you have to do all sort of these filters. You have to do the in silico analysis, you have to do the molecular dynamics, you have to do the MD simulations also to know exactly what is going on. So it's a tedious job. That is the actual challenge what we are talking about, the lead identifications. Okay, if you have this one, that hit, which one is actually working on it, that screening is the big task on it.
So actually I am working on anti-cancer activity. So I got the results of my synthesized molecule.
Okay, but if you say anti-cancer, the first question is coming, which cancer? Now, did you try for this one? And then honestly speaking, there are lot many molecules are having anti-cancer property.
Yes, sir.
But at the end, you see there are how many side effects that you say that no, this I cannot take it as an anti-cancer thing. Okay, so these things are there. So if you are not very, very specific on it, so it is little difficult to actually comment that this might be a drug in this one. No, totally no. Because I can tell you one actual example. One of my friend, they are actually not only working to the anti-cancer molecules, they are basically working to develop the vaccine for cancer. So the T cell, that are stimulator of the T cell, so they will be sting agonist basically. Okay, so they are actually working in such a way that you will be having the the potency in your body so that the cancer cell will not be coming into that. See, he is having all facilities, and as a chemist, we are also some molecules, we can say, okay, we are having the anti-cancer property of my molecules. I talked to him, he is a very good friend of mine. I talked to him several times that you just test the in vitro studies of our compound and give us some data. He is not interested. He said that I am not doing any kind of anti-cancer. My objective is not to develop an anti-cancer drug in synthesized molecules. No. Rather, objective is that which can stimulate the T cell, that is working as a sting agonist, not to have anti-cancer property. So these things are actually there. So you have to be very, very specific on it. Now you can understand how many anti-cancer molecules synthesized by a lab is coming into the market up to now. So so many things in a publications you can write this molecule is synthesized which is having this anti-cancer property, this one. But do you feel that at the end of the day that molecule is coming into the market or some? Why not if that is there? So these are the issues actually. So rather if you can fabricate this work, maybe you can channelize it in other way. You see what kind of ligand that is, what kind of structural things are there, and you can have a screening for all the structures which are known drug. Okay, what kind of structural features you can get with respect to a known drug, and that you can compare then. That molecule is how better than some existing drug. Then maybe you can see the importance of the molecule, rather to say the molecule is having anti-cancer property. Anything? Any other questions? Is that?
Okay, so we'll close this session with a big thank to Dr. Haldar for his insightful talks on synthesis and methodology. Thank you, sir.
Okay. So our next session will start about 12:45. Again, like that, Shiva Kumar, he wrote to me.